Good morning, everybody. I'm very, very happy to have been invited again to this amazing congress with a lot of very, very interesting presentations. I'm also very sorry to not be able to be here live with you today. So I'm at the other side of the Atlantic in France right now. And I recorded this video. I really hope that it's clear enough. But if you have any question or if you want to have any more information about what I said or to have the papers, please just email me and I will answer you. You can also contact me via ResearchGate. I can also send you the papers this way. So I am Morgane Robles. I am a lecturer at the veterinary school in Maison Alfort in France. And I'm also a researcher at the INRAE in France also. So today I will talk about the effects of dietary arginine supplementation on placenta structure and function and foal growth. So what do we know right now in the horse? My presentation will be separated in 3 parts. The first part will be about primiparity in the horse. You will understand the link between primiparity and L-arginine during this presentation. Then I will present you what is L-arginine, its roles and metabolism, and its effects. So the effect of supplementation of L-arginine in the pregnant mares. What do we know so far? Which is not so much, you will see. So as a first introduction, I will talk about the DOHAD concept. So you may have heard about this concept in the previous presentations, the concept of developmental origins of health and disease, but I will still introduce my presentation with that concept. So there are several factors that can affect the maternal environment. You have, for example, the nutrition of the mare, her parity, I will talk about that after, and for example, the obesity. So all those factors, they can affect the metabolism of the mare, her uterine environment, for example. And this will lead to adaptations of fetal development to these modified environments. So these adaptations, they will be made from like epigenetics to the phenotype and of the individual. And one of the most important organ that will affect the fetal development is the placenta. The placenta is considered as the programming agent of fetal development. It will also adapt to the maternal environment. And it's the organ that is involved in the nutrient transfer, the gas transfer, etc., between the fetus and the mother. And it's a very important organ because it's also an endocrine organ. It has a lot of roles. It also has roles in immunity. This is why I will talk a lot about the placenta during this presentation. So that's why I wanted to really insist on the very important role of the placenta in this subject, in this topic. All those adaptations of the fetal development that can lead to an increased risk of developing pathologies at adulthood. So what are those pathologies in the horse? So those pathologies are non-communicable diseases. In the horse, you have the metabolic diseases, such as laminitis metabolic syndrome, Cushing syndrome. You have skin diseases. You have osteoarticular diseases, such as osteochondrosis, digestive diseases, cardiovascular diseases, respiratory diseases, and reproduction diseases. So in the horse, the ones that have been studied are metabolic, osteoarticular, and reproduction diseases. We don't know much about the others, but you can see that just with the changes in maternal environment, you have a lot of diseases that the horses have that are very important in this species that could be prevented. If you're interested in this concept of developmental origins of health and disease, we wrote some reviews on the subject. So if you can't download them or if you don't have access to them, just send me a mail and I will send them to you. So first, the primiparity in the horse. So I will start with just telling you that we recently published a very, very extensive study about female age and parity in horse. So how does age and parity affect fertility, embryo development, fetal development, long-term development of the foals? So if you're interested in this topic, just send me a mail if you can't, if you don't have access to this review, and I will send it to you. It's very, very, very extensive. It's a lot of information. So, primiparity in the horse is considered as the natural model of intrauterine growth retardation. So, what is intrauterine growth retardation? It's when the baby is born in the 10th percentile of weight for its own population. The first 10 percentile means like very, very low birth weight compared to the population. And we showed that primiparity, the foals born to prepose mares, they are born in the first 10th percentile for this population. So they are born very, very, very lighter compared to other horses born to multipose mares. And it's very interesting because all the studies that studied this difference of birth weight between primipose and multipose animals, they showed that foals born to primipose mares are lighter, significantly lighter, compared to foals born to multipose mares. And this difference is in average 15%, which is really a lot, like between 4 and 10 kilos of difference in these studies. And it's always observed independently of the study, the country, the breed. There is always a difference that is very important. And in humans, babies that are lighter, that are born lighter, that are in intrauterine growth retardation, they have more risk of developing non-communicable diseases at adulthood, such as diabetes, cardiovascular diseases, and they also have more risk at dying earlier from those diseases. If we look at the growth of the foals, we can see that foals born to primipose mares, they stay lighter and smaller until at least 12 months of age, compared like dependently of the study. So it's very interesting to see that they are born lighter, but they remain lighter and smaller for a long time compared to foals born to multipose mares. And a study showed also that foals born to prepose male, they have a decreased insulin growth factor 1 serum concentration, which is a marker of growth. It's a growth factor until at least 4 months of age compared to foals born to multipose males. So they're smaller, they remain smaller, and they have a decreased markers of growth compared to foals born to multipose males. We also showed that the foals born to primipose mare had accelerated glucose clearance compared than foals born to multipose mares. So what we did is when we did the intravenous glucose tolerance test, we injected glucose and then we sampled the blood for 3 hours and we measured glucose and insulin. And you can see that the clearance of glucose is increased. It's faster, which means that those foals have increased insulin sensitivity. And another study showed that the insulin sensitivity decreases when the foals grow, when the foals get older. It's because the foals get adapted to a forage-based diet, which means that the foals born to primipose mares, they remain metabolically immature until at least 4 months of age, compared to the foals born to multipose mares. They have a they are less able to adapt to this new forage-based diet metabolically. We also looked at the testicular maturation. So here is the principal component analysis. So here you see the graph of the variables, so all the genes that we analyze, and here you see where the foals they map according to those analysis, so this gene expression. So we analyzed the genes marker of testicular maturity, markers of testicular immaturity, and markers of the establishment of the blood-testis barrier, which we showed earlier that are associated with an immaturity. So there are also markers of immaturity. And you can see that the multipose foals, they cluster with the genes marker of testicular maturity. Well, the primiparous folds, they cluster at the other side of the graph. And especially those two genes were associated with the multiparous folds, while this gene was associated with the primiparous folds. So the conclusion of these results were that the folds born to primiparous males were had less mature testes compared to the folds born to multiparous males at the onset of puberty, so near 1 year of age. We also did a study that had not been published yet, where we compared the performances in show jumping of the horses uh, according to the parity of the mare. So we had access to all the performances of all the horses in France. So it was more than 2,000 hundreds of horses. And you can see that foals born to primiparous mares in orange here have decreased performances compared to the foals born to multiparous mares. It means that the effect that we observed physiologically in the growing foals, we can observe them that affect the performances of these foals in adult age, which is very, very interesting. So what happens during the first pregnancy? Why do these foals are born lighter, they remain smaller, they are immature, they have decreased performances? Why is that? So first, you can see here that the placentas of primiparous pregnancy, they are always lighter at birth compared to the placentas of multiparous pregnancies. It means that there is a decreased, an altered development, fetoplacental development during pregnancy, during this first pregnancy, and a difference in volume is also observed. So here is the placenta of the horse. It is called allantochorion. So in green you have the placenta, and in pink you have the maternal uterus. So there are two tissues that are involved in fetal nutrition in the horse. Those tissues are called trophoblasts. Those are the epithelium of the placenta. You have the hemotrophic trophoblast. So the hemotrophic trophoblast is located along the microcotyledons, so which are those tree-like structures that are very deep, interacting with the maternal uterus. So the hemotrophic trophoblast is called hemotrophic because it's involved in the nutrient exchange from the maternal blood to the fetal blood. And then you have the histiotropic trophoblast. It's located around what we call the areolae, which are a structure that are between the microcotyledons. And this trophoblast is called histiotropic because it's involved in the exchange of nutrients from the maternal uterine glands. So there are two types of fetal nutrition in the fetal horse. In primiparous pregnancies, what has been observed is that there is a decreased surface and or volume of either the histiotropic trophoblast, so that one, and the allantoic vessels, so those ones, or the microcotyledonary vessels, so the ones that are in the microcotyledons, or a decreased volume surface for this paper of the microcotyledons themselves, or of the hemotrophic trophoblast, that one, and the microcotyledonary length. So the results are a little bit different depending on the study, but what we can conclude is that there is a decreased exchange surface of fetal-maternal exchange surface in primiparous pregnancies compared to multiparous pregnancies in the placenta. So the placentas are smaller, lighter, but they also have a decreased exchange surface of the tissues that are involved in the exchange between mother and fetus. When we looked at the functions, we also showed that there is a decreased gene expression of the placental growth factors and their receptors, and also of the factors of vascularization in the placentas from pluripotent mares. And an increased gene expression of genes involved in amino acid transfer and metabolism. Which is interesting is that we also showed that placentas of primiparous mares, that you can see here, the primiparous, and here the multiparous placentas. And here is the amino acids in the no, not the placentas, the mares in red and the foals in green. So you can see that in the blood of mares and foals, in the premipose pregnancy, premipose mares have increased concentrations of amino acids, while the foals have decreased concentrations of amino acids at birth, which means that premipose mares and premipose placentas are less able to transfer the amino acids from the mother to the foal, despite these differences in gene expression that we observe. So there is also an altered function, placental function in those premipose pregnancies. Another thing is that in the premipose pregnancies, you can see that there is a decreased uterine artery diameter and blood flow. So there is a decreased blood exchange between the mother and the fetus in those premipose pregnancies. Also, normally a pregnant mare in early pregnancy will be more able to absorb glucose, and it will increase the glucose storage as fat and glycogen in adipose tissue, liver, and muscles. In late pregnancy, the male will become more insulin resistant. So you can see that there is still an increase in glucose absorption, but not an increase in insulin production. It means that the mares become more insulin resistant. You can see here that insulin sensitivity decreases, and it means that the glucose will be preferentially transferred to the fetus because the transfer of glucose through the placenta is insulin independent. So the mare becomes more and more insulin resistant during pregnancy to transfer preferentially the glucose to the fetus. And at the end of the pregnancy, the mare will use protein and all the fatty acids that she stored in her tissues to sustain her own needs of energy instead of the glucose from the food. And here you have the premipose mares in green and the multipose mares in black. And you can see that the premipose mares, after the meal, they have an increased glucose concentration, but also an increased insulin concentration compared to the multipose mares in late pregnancy. And you can see that the increase of insulin concentration is very high. So it means that the premipose, and also we showed that the premipose mares, they have an increased ability to dispose of the blood glucose in their peripheral tissues. It means that those mares, they are less able to metabolically adapt to pregnancy. They are more insulin sensitive. So they will more store their glucose into their own tissues in late pregnancy instead of sending them to the fetus. So they are less able to adapt metabolically to pregnancy, which could affect the glucose transfer towards the fetus. So in conclusion, for this first part, foals born to premipose males are lighter and smaller, they are less mature, and they are less performing at adulthood. The placentas for premipose males are lighter and less voluminous. They have a decreased surface and volume of the structure involved in the nutrient and gas transfer. They have a decreased expression of growth and vascularization factors, and they have a decreased ability to transfer the amino acid to the fetus. Also, premipose males have an inability to adapt metabolically to pregnancy, which means that they may be able to transfer less glucose to the fetus, and the glucose is the most important nutrient for the fetal development, especially in late gestation. And they also have a decreased uterine artery diameter and blood flow. So the uterus needs to be in some way primed by a first pregnancy before it can achieve its full potential in terms of promoting placental and fetal growth. It's a citation from a paper from Twinkalen and Sandra Wilshire that it published in 2003. So premaritality is a necessary step. You can't have foals without premaritality. It's impossible. There is a first time for pregnancy. But is it possible to use nutritional supplements in pregnancy to improve pregnancy outcome for these first pregnancies. So this is where we talk about L-arginine. So L-arginine is an amino acid. It's considered essential in the horse, especially in gestating, lactating, crowing, and exercise horses. So the horse milk has been shown to have more L-arginine compared to other species, except for the cats. You can see here in this graph that horse milk is very more concentrated in L-arginine compared to other animal milks, which means that L-arginine is more important for the foals than the other species. Also, in vitro, it has been shown that the equine enterocytes, so the epithelial cells of the digestive organs, from young and adult horses are unable to synthesize de novo arginine. So arginine is very important in the horse because they really need it. So currently there are no requirements for L-arginine in the horse. We don't know how much the horses need arginine. There's just one thing that you need to remember is that L-arginine and L-lysine, they compete for the same transporters. And you know that lysine is the most important essential amino acid in mammals in general and in the horse also. So the balance between those both amino acids is very important. It has been shown that the distribution of 120 grams of L-arginine, so for 500 kilos horses, affects the absorption of other amino acids, including the essential amino acids. In our studies, we distributed 100 grams of L-arginine per day. It decreased the basal but not the postprandial lysine plasma concentration. So we did not see any effect on maternal and fetal development of this decreased lysine plasma concentration, and it's only the basal lysine concentration, so in the morning after the whole night. So it could be due to a decreased renal lysine reuptake, we don't know, but it did not seem to affect either the maternal health nor the fetal development. So what are the arginine sources for a body? The first one is an arginine endogenic synthesis from another amino acid, which is not a proteinogenic amino acid, which is a citrulline in the kidneys. So the cells in the kidneys, they can transform citrulline to arginine. Second source of arginine is food, and there is an intestinal absorption. You have to know that arginine is quickly metabolized in the enterocytes and the hepatocytes in ornithine through the urea cycle. So there are other amino acids such as citrulline that may be more interesting to study because they are not transformed into ornithine. But arginine is absorbed through the enterocytes and then will be metabolized into ornithine, preferentially in the liver. You have also protein catabolism that will produce some arginine. Urea cycle does not produce arginine that it can be used by the cells. It's really this cycle, the nitric oxide citrulline cycle, that produces the arginine. So what are the roles of the arginine? So when it's transferred into the cell, the arginine will be transformed into nitric oxide and citrulline through the citrulline nitric oxide cycle. It will also be transformed into ornithine through the urea cycle. It will also be metabolized into creatine and agmatine, putrescine, and polyamines, or it can be used to synthesize proteins. So arginine can be transformed into a lot of different metabolites. And those metabolites have different roles. So putrescine, spermidine, and spermine, so the polyamines, they act as growth factors. Nitric oxide is a potent, and it is the most potent vasodilatator in the mammal cells. It also acts as growth factors and also in proliferation, cell proliferation. Creatine is an anti-inflammatory antioxidant factor, and it also increases insulin sensitivity. And arginine metabolism can also affect changes in DNA methylation because it's involved in the 1-carbon metabolism also. So arginine can affect a lot of different pathways through all its metabolites. And arginine itself has been shown to stimulate insulin production from the pancreas through a pathway that is different from the glucose. So, for example, during the fetal development, when the glucose does not yet stimulate arginine, insulin production by the pancreas, arginine can already. So, why L-arginine and primiparity? So, L-arginine can, we'll see later, increase the blood flow through the production of nitric oxide, so which is a vasodilatory agent. It can also affect fetal development through polyamine and nitric oxide production, which are growth factors. And it may also help mother to adapt to pregnancy through the production of creatine. And you can see that an increased blood flow and an improved maternal metabolic adaptation could also improve the fetal placental development. So this was our hypothesis. So now we'll see if we confirm them or not. So now a review of the studies that studied the effect of supplementation of L-arginine in the pregnant male. So in other species, and especially in the pig, there was the study that has been the species that has been the most studied. So in the pig, L-arginine supplementation during pregnancy increases the litter weight, increases the number of live-born piglets, increases the placental weight of the litter, and stimulates the nutrient sensing pathway and the protein synthesis in the trophectoderm. So the trophectoderm is the trophoblast in very, very early pregnancy when there's still an embryo, so it's not a fetus yet. So there are very encouraging results in the pig that have been observed. So what about for the horses? So for the horses, in early gestation, an oral supplementation of 70 grams of arginine per day for 600 kilos mares. So for very early, from 15 to 45 days after ovulation, has little to no effect on embryo and fetus size. But it's all the authors studied, so we don't know if it has effect on function and structure of the embryo and the fetus. In late gestation, so an oral supplementation with 100 grams of arginine per day from 21 days before falling until 7 days after increases the non-pregnant hormone arterial blood flow, which is very interesting. Shortens the gestational length, but does not affect the fetal placental weight at birth. So L-arginine might improve the arterial blood flow in pregnant males, and we saw that premenopausal males have a decreased arterial blood flow compared to menopausal males. And also supplementation with 50 grams of arginine per day from 19 days before falling until 40 days after does not affect the pregnancy length, however. And during lactation, an oral supplementation with 50 grams of arginine per day during lactation does not affect milk composition nor false growth and adiposity. So the results in the horse are very small, but those studies, they have both multipause and premenopausal males, and they did not study the effect of parity and arginine at the same time. So we first did a pilot study when we had 4 males in the control groups and 4 males in the supplemented groups. So we could not, for this pilot study, really compare the parity. But we wanted to see if the bears took the L-arginine because it has a bitter taste. And if it somehow affected, improved the placental development independently of the parity. So the first result, we showed an increased birth weight of the foals born to supplemented mares, a decreased volume of the histotrophic trophoblast, but an increased volume of the microcotyledon vessels. So those results are very preliminary and needed to be confirmed because we had very like only 4 males in each group. Uh, we also observed a decreased number of degenerative trophoblast cells. So there are degenerative trophoblast cells, so you know the cells that are involved in the nutrient exchange from the mother to the fetus in the placentas at term. And the males that had L-arginine supplementation, they had less of those degenerative cells. So you can see here is the cells that have no, like they look empty, as if they did not have any organoids remaining. Here you can see like a normal cells with the nucleus and other organoids in them. Here they don't have them anymore. And we also saw that supplemented mares had an increased number of microvilli, so those villi, at the apical side of the trophoblast cells. So those microvilli, they are they increase the exchange surface between the maternal side and the fetal side of the placenta. We also here you can see it's so here it's microscopy, electron microscopy picture, and here you have the transmission one, and you can see that here it's a control. You can see that there are cells with no microvilli, like entire cells with no microvilli at the apical site. And in the arginine group, you have way less cells that have no microvilli at the apical site. And we also made a count of the number of vesicles in the endothelial cells and along the basal lamina. So the more vesicles that you have, the more cell activity you have. And we observed an increased number of vesicles in the arginine group compared to the control group. So as I said, those results are very preliminary and we did not publish them. But we continued with a study that we published after. So this study. It was published in Scientific Reports, so it's open access, so everyone can have access to it. What we did is that we supplemented 7 multipose and seven plumipose males with arginine, and as a control group we had 7 multipose and 6 plumipose males that were not supplemented. So the males were in pasture until 4 months of pregnancy. So they had free access to water, mineral, and vitamin supplements. The pasture are in the center of France, and the mares have a lot of places like grass is really, really sufficient for them. From the 4th month of pregnancy, so it's autumn, the mares were back to the barn and they were fed hay, haylage, and flatten barley mixed with minerals and vitamin supplements, so which were enough to bring enough energy and proteins and fibers to them. And from 210 days of pregnancy, we supplemented the mares until falling. So it's a supplementation during late gestation that we did. So here is what the mares ate according to their body weights. So in black, you have the multipose mares control, in green, the plumipose controls, and in blue, the plumipose arginine. And you can see that the mares that received L-arginine, they really received way more L-arginine than the other males. And that plumipose males, because they were a little bit lighter, they received a little bit more lysine and a little bit more of energy compared to the multipose males during their pregnancy. But it did not affect their body condition and their body weight during the pregnancy. So the difference is significant here, but it did not affect the mirrors more than that. So when we studied the amino acid concentration in the blood of the mares, you can see that what I said is that the pluripotent arginine mares, they had a decreased basal lysine plasma concentration. So in the morning before we feed them, but we did not see any other effects on lysine concentration. And as I said, it might be a decreased lysine reuptake in the kidneys and not a difference in absorption, because we did not see any difference in lysine concentration after feeding, so in the postprandial concentration. So it might be a difference in renal reuptake, but we did not see a difference in absorption of lysine between the supplemented and the control groups. You can see that the pre-pause mares that were supplemented with arginines have an increased arginine blood concentration, as well as an increased ornithine blood concentration compared to the other groups. So arginine was expected, and ornithine also, because I told you that arginine is quickly metabolized into ornithine in the hepatocyte. So this is what we expected. When we look at the plasma, so the amino acid concentration in the plasma of the males and the foals at birth, you can see that there is no really, like, the most differences between multipause and pluripause males, but arginine did not really affect the concentration of amino acids in the males and the foals at birth. The only amino acid that differed was the ornithine, that was increased in the blood of the supplemented mares at birth, but no effect in the blood of the foals. So L-arginine supplementation increased the postprandial maternal plasma arginine and ornithine concentration, but did not affect the fetal amino acid concentration at birth. We then looked at the effect on metabolism in the mares. And I showed you this graphic already, and I showed you that the pluripotent mares, they had increased glucose and insulin concentration after feeding. And what is very interesting is that the pluripotent mares in blue, you can see that their curves after feeding are very close to the curves of the multipose mares. And we also did the glucose tolerance test, and we showed here, so remember, the pluripotent mares have an increased ability to dispose of blood glucose into peripheral tissues, and arginine supplementation decreases this ability and bring it closer to the one of the multipose males. So it looks like L-arginine improves the maternal metabolism in late gestation for those pluripotent mares. It helped them to adapt metabolically to the pregnancy. We then looked at the structure of the placenta, and we did not see any difference between the control and the supplemented groups. When we looked at the function, also the gene expression, you see that there is an increased expression of the glucose transporter, which is very interesting because we saw that the males have an improved ability to adapt to glucose, to metabolic, and increased metabolic ability, to adapt to the pregnancy. And it shows here also that they may be more able to transfer the glucose from through the placenta to the fetus. We also observe an increased expression of fatty acid transporter and an increased expression of this enzyme, which is involved in the creatine production. So there may be this enzyme, which is involved in creatine production. So there may be an increased creatine production in those placentas. So L-arginine supplementation increases the placental gene expression of glucose and fatty acid transporter, and maybe of creatine production. So it's maybe more involved in this metabolic adaptation to pregnancy. When we looked at the methylation, DNA methylation, because I told you that L-arginine supplementation might also affect methylation, DNA methylation in the placentas, you can see that the methylation in primipost males is decreased in the placenta. So primiparity affects the epigenetics of the placenta compared to the multipost males. The supplemented placentas, you can see that it's in between both groups. So arginine may increase the DNA methylation, but not enough to be different from the primipost, but also it's not different from the multipers anymore. So it might affect the methylation, but we need to study more this methylation to see where and how it affects it. And for the full birth weight, we did not see any difference in full birth weight, but we had a very, very small amount of horses. So maybe with more horses, we would see a difference. So in conclusion, for this study, L-arginine supplementation in late gestation increased the maternal postprandial arginine and ornithine plasma concentration, but did not affect the amino acid transfer to the foals. Improved the metabolic adaptation to pregnancy of primipost males, which is the most important result that we observed here. Increased the placental expression of the glucose and fatty acid transporters, but did not affect the placental structure, and appears to be harmless to both males and the foals. So what I have to conclude is that those are preliminary studies. You see that there are not so many studies of the effect of L-arginine supplementation in the horse, and we don't know much. We also don't know much about the maximum quantity that you can give to a mare and the most effective one. So all those results are very interesting and they are encouraging, but right now we can't really confirm or conclude about how and how supplementing L-arginine, the concentration and when, and if it has an effect, a long-term effect on the foals. We really don't know that. So it's interesting, but there are needs for more studies on this subject. So as a general conclusion of my presentation, I want you to remember that primiparity affects the fetal and long-term development in the foals, but can't be avoided. Little is known about L-arginine requirement, metabolism, and safety in horse species. And the first results are encouraging, but more studies are needed to understand the short and long-term effects of L-arginine supplementation of the pregnant mares. So I want to thank you for your attention, and I want to thank all the people that worked with us for these studies, and especially Pascal Chavat-Palmer, which is the head of my lab, and Emilie Derizou that worked with the maternal age and parity and also worked on this paper on L-arginine supplementation, as well as Audrey Giverding that did all the analysis for the histology, the placental histology analysis. And again, thank you for your attention, and I hope you have, I hope you understood everything. And as I said, if you have any question, just send me a mail and I will answer you. Thank you and goodbye.